Welcome to Knowledge Domain, Concept of Life Sciences and Biotechnology. In continuation with the plasma membrane models, today we are going to discuss unit membrane model, also known as the greater membrane model, proposed by Robertson in 1957. Robertson was working on frog peripheral nerve fiber. He is a electron microscopy. He was using osmium tetraoxide and potassium permanganate as a fixating agent. This is used for the sample preparation to be studied in the electron microscope. And the dehydrating agent is the acetone. He proposed that plasma membrane and membranes of all organelles are similar. He called it unit membrane because all membrane, the thickness is almost 75 angstrom, and as you can see in this electron micrograph that it looks like a railroad where two parallelly dense lines are separated by a light band. And in this unit membrane, we can see here, these are the protein layers, which are the mucoprotein that is present on the outer side of the plasma membrane, that means the extracellular environment. And these are the proteins found in the intracellular side of the plasma membrane are the non-nuclear proteins. And to the extracellular side, you can see the glycoprotein backbone, oligosaccharide chain, and the sialic acid terminus. In this case, the protein part is almost 20 angstrom in thickness, and the phospholipid is almost 5 angstrom in thickness. So 25 here, 25 here, and this phospholipid bilayer, only the lipid, they constitute almost 25 angstrom in the thickness. And the lipid bilayer with protein monolayers are covalently attached on the both sides. Lipid and the proteins are covalently attached on the both sides, and the composition is 40% lipid by weight. And as the first time it was proposed that the plasma membrane is made up of the lipids, this unit membrane model shows high electrical resistance and high permeability to the non-polar molecules and impermeable to the small ions. The Jostren in 1963 did not agree with the unit membrane model, and he argued that the mitochondria, endoplasmic reticulum, and the plastid membranes are not like the plasma membrane and they display particulate units. So the same unit membrane model can also be explained as the trilaminar model. Benson in 1966, he was working on the chloroplast membrane and he found the lipid and the proteins form a hydrophobic association. The charged polar heads of the phospholipid molecules lie on the surface of the membrane and are capable of binding ions, which are hydrophilic in nature. The non-polar hydrophobic lipid tails are linked with the complementary hydrophobic regions within the interior of the protein, as you can see the unit membrane model. Larnard and the Singer in 1966, they argued or they proposed that one-fourth of the proteins are in the helical conformation and the rest from the random coils. Protein crystal model was proposed by the David Green and the co-workers in 1970, where protein polymerized to form two layers of loosely packed globular protein units of 30 to 40 angstrom in diameter, like here. These are the proteins, and each of them is almost 30 to 40 angstrom in diameter, and they are present in the two layers. And the combined thickness of the plasma membrane using the two layers is 70 plus minus 10 angstrom, as we can find in the all the membrane proposed till now. That plasma membrane is almost 75 angstrom in diameter. And a non-polar region may exhibit the hydrophobic bonding with the non-polar group of the phospholipid molecules. Non-polar hydrophobic interactions that fill the cavities between the globular protein units, while the polar heads of the phospholipid molecule face the membrane surface. So this is the polar head, they face the membrane surface, and this is the Hydrophobic, they fill the gap between the protein molecules.